Welcome back. You are at Anatomy and Physiology 2, Bio 212 at Norwalk Community College. We are finishing up Chapter 18, and by that I mean we have introduced the endocrine system. We have considered the fact that the molecules that travel through the circulatory system by being released into the interstitial space are either water-soluble or water-insoluble. And we've spent a fair amount of time regulating the expression of these so that you understand that you have either releasing hormones or inhibiting hormones that are made inside the hypothalamus that target the anterior pituitary gland, where you have 5 populations of cells: somatotrophs, lactotrophs, corticotrophs, thyrotrophs, and I can't think of the last one. Let me see if I can look it up for you here quickly. Last population of cells. There they are. Corticotroph, lactotroph, gonadotroph. That's what it was. And the significance, of course, is that by regulating these, we can then make a series of hormones. And what we're doing now is we're moving on to how it is we actually regulate glucose levels. And we're going to target primarily the pancreatic islets inside of the body. The pancreas itself is located posterior and inferior to the stomach inside your body, and it has a specific responsibility. It has both exocrine and endocrine responsibilities. The exocrine responsibilities we come back to, or we cover inside the digestive system, right? When we're actually making materials to mix inside the small intestine. We're thinking primarily now about the endocrine responsibilities, which means 1% of the remaining cells make up these things that are called pancreatic islets, or the islets of the Langerhans. Okay? And there are 4 populations of cells inside of them: alpha, beta, delta, and F cells. And what's important for you to understand is that alpha cells are going to be instructed to release a specific molecule, and that's called glucagon. And glucagon will raise blood sugar. In a different set of signaling situations, beta cells will be instructed to release insulin. And insulin lowers blood sugar levels. Delta cells we come back to inside chapter 24, but for the most part, somatostatin, which is what the D cells make. D cells make and secrete somatostatin, and they inhibit both insulin and glucagon. And then finally, F cells secrete pancreatic polypeptide that inhibit somatostatin. So F cells shut off D cells, which means then A cells and B cells can do their job. They also incite gallbladder contraction, and they incite the release of pancreatic digestive enzymes. So you could see how, if we had time and inclination, and your textbook doesn't do this, although maybe someday it will, Mother Nature has put in one small area all of these cells that are acting antagonistically to one another. Alpha cells and beta cells raising and lowering blood sugar. Somatostatin and pancreatic polypeptide inhibit one of these functions. So somatostatin inhibits both A and B here. F cells inhibit D cells so that A and B can function. Just something to think about. Okay? Well, why is it that we actually need to think about this? So I'm going to go to this figure here. First, I need to erase all of that drawing. I'm going to go to this figure here. And here is an islet of Langerhans, or a pancreatic islet. And you're going to see it has a how do I say this? It doesn't have a uniform border, or margin would be what we would call it, right? There's no uniform margin. Because there's no uniform margin, it's contrasted against these acini cells. These acini cells are the exocrine function, and there are millions and millions and millions of these. And notice they have this sort of rosette formation, but we're not interested in them today. It's inside here that we actually have those A cells, B cells, A. And I'm not pointing to anything, whoops, that line has to come all the way over here. I'm not pointing to anything specific here. I just want to make sure you get the idea. B cells, delta cells, and F cells, right? And over there, they're listed out for you. Remember, this is just a cartoon. There's no way you actually see it like this in reality. In fact, you can't even tell them apart when you're looking here, right? What I want to make sure you understand is that B cells actually measure the amount of glucose inside your bloodstream. So B cells actually have multiple functions. And if the glucose level gets to be too high or too low, insulin can actually do something here. Obviously, if glucose gets to be too low, glucagon is going to be made. And glucagon is going to travel to primarily the liver, where at the liver it will cause the hydrolysis of glycogen and glycogen, which is the polymer, right, of glucose, to release that into the bloodstream and therefore increase glucose levels. Okay. If we increase insulin levels, though, insulin actually binds. Here's a cell with a receptor on it. Insulin is the blue dot. The blue dot binds the insulin receptor, and what happens now is you increase the number of transporters, which are called GLUT transporters, glucose transporters, and now you can get glucose into the cell. So what that means to the actual glucose in the circulatory system means the concentration now has dropped. Remember, this is somatostatin, pancreatic polypeptide. These regulate the effects. So if somatostatin stops A and B, remember F stops D. Okay. So let's take a look at that, meaning the A and B part. In this case, you actually have low blood glucose level, which means in this case, alpha cells secrete glucagon. Glucagon travels to those liver cells, and what's going to happen is you're going to take the glycogen and we're going to release that as glucose into the bloodstream, okay? Now, the form of glucose, and what we mean by this, of course, is that you can actually, with glucagon at the same time, you can convert glycogen into that glucose, no problem. We can just lyse that. But we can also take from lactic acid and certain amino acids, we can form glucose, and that actually has a specific name. That's this is gluconeogenesis. More important for later in the semester, but this is where this idea is introduced. So in times of plenty or in times of want, you can use lactic acid, which is the waste material for glycolysis, and or certain amino acids. That glucose is released by the hepatocytes, and then we return it back to its normal concentration. Remember earlier, I gave you a figure that looked something like this, but we used different hormones. We used human growth hormone, right? Now we're looking at insulin and glucagon. Now what's going to happen, of course, is you're going to have blood glucose level rises. It's going to be it actually inhibits the release of glucagon. Insulin over here, though, these are made by beta cells. Beta cells release that insulin. Remember, I told you a moment ago, here's a drawing of that cell. Let's see if I can make this one a little bit better. Cell, I'm going to leave a little channel there so I can put a glucose transporter there. Glucose transporter. But remember, for the glucose transporters to work, we have to have insulin, binding the insulin receptor. We increase the number of glucose transporters inside of the cell, and that means, I'm going to try and draw insulin better here. This six-sided structure is transported in here, and then we're going to phosphorylate it to make sure that it doesn't come right back out, right? Glucose 6-phosphate, right? Now, insulin acts on various body cells inside your body, okay? It accelerates the facilitated diffusion of glucose into the cells, just showed you there. It also has a conversion of glucose into glycogen, okay? So that means insulin can target the liver and or skeletal muscle tissue so that you actually store glucose as glycogen. It also causes the increase in the amount of amino acids, amino acid, AA, increase uptake into the cell, that's the nucleus here, okay, as well as protein synthesis, which means in the presence of insulin, we can increase protein synthesis using those amino acids that have just been taken in. It also speeds the synthesis of fatty acids, which is called lipogenesis. It slows a glycogenolysis, and it slows gluconeogenesis. So it acts antagonistically to the mechanisms of glucagon we see here. Blood glucose levels will fall, and if this continues, then we actually have hypoglycemia, and then we start the whole thing all over again by releasing glucagon so that alpha cells, okay, will cause the release of glucose from those stores of glycogen. So that's probably the most important idea at this point in time, and that is we actually have a mechanism here to compare and contrast antagonistic relationships at the endocrine level. It requires energy, meaning this is the basic mechanism that allows for energy to be metabolized inside of the cell. So even though this looks like a pretty straightforward analysis, I want to make sure you're paying attention to the fact that, hey, without insulin, we don't get glucose into cells. If we don't get glucose into cells, we do not survive. Okay. Your textbook now switches over temporarily or quickly to some extraneous hormones inside the body. Okay. And the next set are the ovaries and the testes, right? This is actually how we differentiate between males and females. These are the gonads that produce gametes, and the gametes are, of course, meiotic cell products, which means they have the normal half of the assortment of chromosomes after they've been recombined. So if a normal cell, somatic cell inside the body has 23 pair of chromosomes, all gametes have 23 chromosomes, and they are haploid. The gonads also produce hormones, and those hormones are specific to the sex. Ovaries produce two estrogens, estradiol and estrone, okay, as well as progesterone. And we are going to regulate how they function. You spend more time inside chapters 27 and 28 and 29 rather, figuring out how all of the gonad functions and the hormones that are made regulate from fertilization, meaning the ability for a gamete to be made available, to birth, okay, can happen through the regulation of these three hormones as well as these accessory hormones down here. And then testes for the male, they regulate testosterone production, and testosterone production regulates sperm production, which maintains the likelihood that fertilization is going to happen with that secondary oocyte over here, but also the male secondary characteristics, which is going to facial hair, Adam's apple, things like that. It also has another hormone, and that's called inhibin, and inhibin is going to release follicle-stimulating hormone. And before I go on, what I want to do is I just want to draw. Actually, you know what I'm going to do? I'm going to go all the way back to the beginning here. I'm going to go right here. I'm going to go right here just to make it easier to understand. Fantastic. So for both males and females, remember, some stimulus is going to be detected inside the brain, and it's going to cause, in this case, gonadotrophic releasing hormone. Gonadotrophic releasing hormone enters into the hypophyseal portal system, which then delivers it to the gonadotrophs. And the gonadotrophs here, regardless if it's male or female, release FSH and LH. FSH and LH are follicle stimulating hormone and luteinizing hormone. Okay? Each will have a different responsibility in males and females. But it's important to understand that for males and females, in the hypothalamus, we have gonadotrophic releasing hormone, and that's now going to cause the release of FSH and LH. Okay? So far we've covered the somatotrophs, thyrotrophs, gonadotrophs, corticotrophs. We don't cover lactotrophs until we get to chapter 28 and 29. But you know what's going to happen. After birth in females, there's going to have to be a regulation that will allow for the prolactin releasing hormone to activate lactotrophs so that you can actually make prolactin, which will then cause breast tissue to actually secrete milk. Okay. Oops, let's get rid of those over there. Hopefully that helped. I mean, I could draw that, but I'd be afraid that, you know, the picture wouldn't come out right and all sorts of stuff could go bad. Okay, let's get out of here. let's go here, the pineal gland. So here was our cartoon of the brain from the beginning of the lecture. There's the cerebellum. Here's the cortex of the brain. And over here is going to be infundibulum, pituitary gland, anterior and posterior, right? There's the pons, there's the medulla. And here's our diencephalon. Our diencephalon has a central thalamic mass. We have our pineal gland, which is also the epithalamus, and here is our hypothalamus. Great. It's attached to the third ventricle of the brain at the midline. Okay, it's a mass of neuroglia and pinealocytes, and the major hormone is an amine hormone, and it's called melatonin. It's derived from serotonin. Serotonin and melatonin actually act antagonistically to one another, and what it does is it sets your circadian rhythm, and that's because when your eyes are closed, eyes, C-L-O-S-D, oops, closed, eyes closed, you increase melatonin. That's supposed to say increase here, E-A-S-E. Not melatonin. Melatonin. Fantastic. Okay. The serotonin is increased during the day because your eyes are open and UV radiation causes the production of the amine hormone, serotonin. Okay. So, pretty straightforward idea. Let's think what happens when we look at, say, the thymus. Point to your thymus. If you just point it to your throat, leave. Okay, the thymus is between the sternum and the lungs, produces thymosin, thymic factor, thymopoietin. We cover this in a few chapters. Okay, they're all involved in T cell maturation. T cell is a specific type of lymphocyte. There are two types of T cells by way of thinking about the future. There are CD4, which are helper T cells, and CD8, which are the cytotoxic T cells. They came, meaning T cells came from the bone marrow to the thymus to mature. Okay? That's why you have all those hormones there. Now, the interesting thing here is, inside your textbook, this is where they actually put the iodination picture, which means this should go all the way back at the beginning. So this is a way for us to actually review. As I'm finishing up this entire system, I'm going to draw a cartoon. Meaning the endocrine system. And I'm going to use the thyroid gland as an example, right? So let's draw you, right? I'm going to put your nose over here and inside your brain, and here's your body, your arms, your legs. Cool, you're supposedly in correct anatomical position, but your head is facing sideways, so it's turned laterally towards the left. Put a smiling eye on you. And I want you to pay attention to the fact that I'm drawing here a brain, and the brain has a hypothalamus. The hypothalamus is going to be sending a hormone, right? So we have, if we need T3 and T4 inside the bloodstream, right? We need T3 and T4 here. What has to happen is thyroid releasing hormone, TRH, has to target the anterior pituitary gland, because inside of here you have thyrotrophs, thyrotrophic cells, and those thyrotrophic cells, when stimulated by thyroid releasing hormone, okay, increase the amount of thyroid stimulating hormone. And that thyroid stimulating hormone is now going to enter into the remaining part of the hypophyseal portal system, and it's going to target. This is supposed to be your thyroid glands. They're hopefully not this big, right? In you, okay, which means you have no iodine. But the point is this: you actually have thyroid stimulating hormone, which is targeting the cells here to increase the production of thyroglobulin. That's what TGB stands for, thyroglobulin. Simultaneously, you're taking an iodide here. It's being trapped, and it's being sent into the inner portion here, that colloid portion. And we're going to add it to the TGB. We're going to oxidize it first. we're then going to iodinate the tyrosines on the TGB. So we're going to start coupling those, and you're going to see the I's are added, and you got 1, 2, 3, 4. Finally, you have 3 or 4 of them. When they get to a specific profile, they're pinocytosed and digested inside the lysosome, inside the cell. This is where you actually make T3 and T4. And now remember, it's a thyroid hormone, so it's insoluble inside of the plasma of your blood. So we actually have to use these binding proteins. They're called thyroxine binding proteins. They were made inside your liver, and now you can actually secrete this through the rest of your body. Remember, T3 and T4 do a lot of things, but essentially the most important is they increase basal metabolic rate and the numbers of sodium potassium ATPases. And A, that's an N, not a W. And A plus potassium ATPases. ATPases. Now my challenge to you, now that you've finished chapter 18, is can you do this and now shut off the system? Meaning, can you go from releasing hormone to the production of the hormone that stimulates the formation of the hormone that we need to use, figure out its concentration, does specific actions throughout the body, namely increased basal metabolic rate, sodium potassium ATPases. What else is it doing inside the body? And then how does your body shut it off? Because it doesn't want to be making this all the time. Can you do that for all of the hormones we've covered now inside chapter 18? Okay? It's a pretty darn good place to stop. Have a wonderful day, and I'll see you guys in class. Bye.